The water rippled as the morning light danced across Lake Baikal's surface. Dr. Elena Zaretsky peered over the edge of the small research vessel, her eyes fixed on the device lowering into the crystal-clear depths. Behind her, Professor Richard Yeung adjusted the sonar equipment, his weathered hands moving with practiced precision. "For 1,000 meters," Elena murmured, marking the depth on her tablet. "We are directly above the trench now." Richard nodded, his focus unwavering. "Oxygen levels are still reading normal. Temperature gradient is exactly as predicted." He paused, tapping a series of commands into the system. "If our theories are correct, we should find them congregating near the hydrothermal vents." Their quest had brought them to the deepest lake on Earth, a 25-million-year-old rift valley in Siberia containing 1/5 of all unfrozen freshwater on the planet. But they were not here to study the lake itself. They sought something far more improbable, a creature that, according to every established principle of biology, chemistry, and physics, should not exist. The remote camera descended further, illuminating the darkness. Both scientists fell silent as the feed displayed on their monitors. The bottom of Lake Baikal came into view, a landscape as alien as another planet. And then they saw it, translucent, pulsing with an inner bioluminescence, moving with deliberate purpose across the lakebed. "Impossible," Richard whispered, but the evidence floated before them on the screen. Elena's voice was steady as she documented the moment. "Desulforudis audaxviator, confirmed present in Lake Baikal at 0942 hours, June 12, 2023. The impossible animal had been found." What made this discovery so extraordinary was not merely the creature's existence in this remote lake, but its very nature. Desulforudis audaxviator, the bold traveler, represented a biological paradigm that shattered conventional understanding of life's requirements. First discovered in 2008 by a team led by microbiologist Dylan Chivian in the Mponing gold mine in South Africa, audaxviator was found thriving 2.8 kilometers beneath Earth's surface in complete isolation from all other forms of life. Unlike virtually every other known organism on our planet, these bacteria did not depend on sunlight or oxygen. They existed entirely alone, comprising a complete ecosystem of one species. This discovery fundamentally altered our understanding of what conditions could support life. The bacteria derived energy from the radioactive decay of uranium in the surrounding rock. They extracted carbon from dissolved carbon dioxide, nitrogen and phosphorus from surrounding rocks, and other essential elements from the mineral-laden water in their environment. Most remarkably, these organisms repaired damage to their DNA caused by the radioactive environment using genes that appeared to have been acquired through horizontal gene transfer from other organisms, before becoming completely isolated millions of years ago. This isolation was so complete that scientists estimated they had been separated from all other life forms for between 3 and 25 million years. The existence of audaxviator in the depths of Lake Baikal presented a scientific conundrum of the highest order. While the lake is ancient, it is not a closed system like the deep mine shafts where the bacteria were first discovered. How had this organism maintained its isolation? How had it adapted to the specific chemistry of the lake? And most puzzling, how had it evolved its extraordinary new capabilities? These questions dominated Elena and Richard's research, funded by a joint initiative between the Russian Academy of Sciences and the International Astrobiology Institute. Their expedition represented the culmination of 3 years of theoretical work following initial water samples that contained DNA fragments matching the unique genomic signature of audaxviator. "We need to collect specimens," Elena said, already preparing the remote sampling equipment. "The real work begins now." Richard nodded, his eyes never leaving the screen. "If the preliminary DNA analysis is accurate, these are not identical to the South African population. They have diverged significantly, which means either parallel evolution or Or they have been isolated from each other for far longer than we initially estimated. Richard completed her thought. Possibly predating the formation of Lake Baikal itself. The implications were staggering. If these organisms had been evolving independently since before the lake's formation 25 million years ago, it suggested a subsurface biosphere of potentially global proportions. A shadow ecosystem operating under completely different rules than the surface biosphere dependent on photosynthesis. The sampling arm extended from the research vessel, collecting specimens with precision. Within hours, Elena and Richard would begin the painstaking process of analyzing these extraordinary organisms, creatures that survived without oxygen, without sunlight, in extreme pressure, deriving energy from chemical processes that would be toxic to most life forms. As they worked, neither scientist fully grasped how profoundly this discovery would reshape humanity's understanding of life's tenacity and versatility. The ability of audaxviator to survive in conditions that would kill virtually any other organism on Earth rested on a series of remarkable adaptations. Most multicellular organisms rely on oxygen to generate energy through cellular respiration. Oxygen serves as the final electron acceptor in the electron transport chain, enabling the production of adenosine triphosphate, ATP, the energy currency of cells. Audaxviator, however, had evolved an entirely different energy production system. Rather than utilizing oxygen, these bacteria employed a process called radiolysis, wherein they harvested energy from the radioactive decay of uranium, thorium, and potassium in surrounding rock. Water, when exposed to radiation, breaks down into hydrogen peroxide and various other compounds. Audaxviator could then use these compounds in a complex series of chemical reactions to generate ATP. The metabolic pathway is extraordinary, Elena explained 3 days later, as she reviewed the latest genomic analysis in the makeshift laboratory aboard their vessel. They have genes for obtaining energy from sulfate, iron, and hydrogen, but the Lake Baikal population shows additional adaptations for utilizing methane seeps unique to this environment. Richard leaned closer to examine the data. That explains their congregation near the hydrothermal vents. They are exploiting multiple energy sources simultaneously. This metabolic flexibility represented an evolutionary marvel. Most organisms specialize in one primary energy acquisition strategy, but audaxviator had developed multiple redundant systems. If one energy source became scarce, they could seamlessly transition to alternatives. Even more remarkably, the Lake Baikal population showed evidence of being able to enter a state of suspended animation when environmental conditions became unfavorable. Their metabolic rate could decrease to near zero, with essential cellular functions maintained at the bare minimum required for survival. In this state, they could persist for what the data suggested might be thousands, perhaps even millions, of years awaiting more favorable conditions. This ability to exist in a state between life and non-life challenged fundamental definitions of what it meant to be alive. Traditional biology defined life as maintaining homeostasis, having organizational structure, metabolism, growth, adaptation, response to stimuli, and reproduction. Yet during their dormant phase, audaxviator suspended nearly all these functions while maintaining the potential to reactivate them. It is almost like they can pause time from their perspective, Richard noted, his voice betraying rare emotion. They exist on a completely different temporal scale than we do. The genomic analysis revealed another surprising adaptation. Unlike most bacteria, which exchange genetic material through horizontal gene transfer when in contact with other bacteria, the isolated audaxviator had evolved an unprecedented capacity for self-directed genetic modification. Look at this, Elena pointed to a section of the genome display. They have developed a novel mechanism for controlled mutations in specific genes in response to environmental pressures. This represented a radical departure from standard evolutionary processes. Rather than relying solely on random mutations filtered through natural selection over generations, Audaxviator could actively reshape portions of its genome within a single generation when faced with new challenges. It is almost like directed evolution, Richard said, but occurring within a single organism rather than across a population over time. This capability had profound implications. In essence, audaxviator possessed a form of genomic plasticity that allowed it to engineer solutions to environmental challenges in real time. When conventional evolutionary processes might take thousands of generations to develop an adaptation through random mutation and selection, these bacteria could potentially develop novel traits in a single generation through targeted genetic modification. The mechanism appeared to involve sophisticated DNA repair systems coupled with controlled mutagenesis. When environmental conditions changed, specific regions of the genome, particularly those involved in metabolism and stress response, experienced higher mutation rates, while essential housekeeping genes remained protected from change. It is like they have partitioned their genome into conservative and experimental regions, Elena observed. Essential functions remain stable, while genes involved in environmental adaptation are allowed to evolve rapidly under stress. This discovery had implications far beyond a single species of bacteria. It suggested that life's adaptive capacity was far more sophisticated than previously understood. Evolution was not merely a passive process of random mutation and selection but could include active, directed components in response to environmental challenges. As the research vessel gently rocked on Lake Baikal's surface, Elena and Richard continued their analysis, unraveling the mysteries of an organism that reimagined the boundaries of life's capabilities. 3 weeks into their expedition, a new discovery further complicated the already extraordinary picture. Using advanced microscopy techniques, Elena identified structures within audaxviator that defied conventional bacterial morphology. "Richard, you need to see this," she called. Adjusting the focus on the electron microscope, he joined her at the station, peering at the images. "That cannot be right. Those look like membrane-bound organelles," Elena confirmed. "Similar to eukaryotic cells, but with completely novel structure and apparent function." This observation contradicted a fundamental principle of microbiology. Bacteria, as prokaryotes, were defined by their lack of membrane-bound organelles, structures like nuclei, mitochondria, and chloroplasts found in the more complex eukaryotic cells of plants, animals, and fungi. Yet audaxviator displayed distinct compartmentalization. Further analysis revealed these structures served specialized functions, including concentrated areas for DNA repair, energy production, and even a primitive form of information processing that resembled neural network activity. This challenges the entire prokaryote-eukaryote dichotomy, Richard said, running his hand through his gray hair. They represent either a transitional form we have never encountered or a completely different evolutionary path. The emergence of cellular compartmentalization represented one of the most significant evolutionary transitions in Earth's history, occurring approximately 2.7 billion years ago and eventually leading to all complex multicellular life. Finding bacteria with similar structures suggested either that this transition had occurred multiple times independently, or that audaxviator preserved cellular characteristics from before the divergence of prokaryotes and eukaryotes. "We might be looking at a living fossil," Elena suggested. "A glimpse of cellular organization from the early Earth, preserved in isolation for billions of years." This possibility aligned with the extreme age of Lake Baikal and the geological stability of its deepest regions. The lake sat atop one of the most ancient and stable continental plates, relatively undisturbed by the tectonic upheavals that had reshaped much of Earth's surface. The mysteries of audaxviator extended beyond its internal structure to its relationship with its environment. Continuous monitoring of the lake bottom revealed that the bacteria created and maintained microhabitats around themselves, altering local chemistry to optimize conditions for their survival. "They are not merely adapting to their environment," Richard noted after reviewing a month of data. "They are actively engineering it." This environmental modification occurred through the secretion of specialized compounds that precipitated certain minerals while dissolving others, effectively creating a protective microenvironment with precisely controlled chemical composition. In some cases, multiple bacterial cells collaborated to form larger structures that optimized the flow of nutrients and energy resources. It resembles primitive multicellularity, Elena observed, or at least coordinated behavior that transcends individual cellular boundaries. The ability to modify one's environment represented a critical evolutionary advantage typically associated with more complex organisms. Humans built shelters, beavers constructed dams, and birds fashioned nests. Finding similar behavior in single-celled organisms challenged assumptions about the cognitive requirements for environmental engineering. As their research continued, Elena and Richard found themselves confronting questions that transcended traditional disciplinary boundaries. The audaxviator was not merely a biological curiosity. It represented a fundamental reconsideration of life's essential parameters. The bacteria's existence in Lake Baikal also raised questions about how it had arrived there. The two most viable hypotheses both challenged conventional understanding of Earth's biosphere. The first possibility was that the audaxviator had always existed in a global deep subsurface biosphere, with populations evolving independently in different locations. This would mean that beneath our feet existed an entire shadow ecosystem, operating independently from surface life, potentially extending kilometers into Earth's crust. The second possibility was even more provocative, that the bacteria had been transported between deep environments, perhaps through groundwater flows over geological timescales or through other mechanisms not yet understood. This would imply a level of microbial dispersal and colonization capability far exceeding current models. Either explanation required a substantial revision of how scientists understood Earth's biosphere and the distribution of life within it. The traditional view of a thin veneer of life on Earth's surface, dependent on sunlight, was giving way to a more complex picture of a planet permeated with life at multiple levels, operating on different energy sources and evolutionary principles. As their research vessel departed Lake Baikal after 6 weeks of intensive study, Elena and Richard carried with them not just specimens and data, but a profoundly altered understanding of life's possibilities. The audaxviator's remarkable adaptations extended beyond its cellular structure and energy metabolism to include unprecedented mechanisms for surviving environmental extremes. Laboratory analysis of the Lake Baikal specimens revealed capabilities that pushed the known boundaries of biological resilience. One of the most striking discoveries came during experiments testing temperature tolerance. Most organisms have a relatively narrow temperature range within which their biochemical processes function properly. Enzymes, the protein catalysts that facilitate cellular reactions, typically denature, lose their functional structure at high temperatures and become ineffective at low temperatures. The audaxviator, however, displayed functional activity across a temperature spectrum from near freezing to over 120 degrees Celsius, well above the boiling point of water at standard pressure. This range exceeded that of even the most extreme thermophiles previously documented. Their proteins must have some novel structural properties, Elena noted during a conference call with colleagues at the International Astrobiology Institute 6 months after the expedition. The tertiary structure remains stable under conditions that would denature conventional proteins. Further analysis revealed that the audaxviator possessed not one but multiple sets of enzymes optimized for different temperature ranges. When conditions changed, the bacteria could express the appropriate set while suppressing others, essentially swapping out their entire metabolic machinery to match environmental conditions. Even more remarkably, these bacteria could survive complete desiccation, the removal of all cellular water, for extended periods. When rehydrated after months in a completely dry state, they resumed normal metabolic function within hours. They enter a state of anhydrobiosis, Richard explained in his presentation to the American Society for Microbiology. All metabolic processes cease, Membranes are stabilized with specialized sugars, and DNA is protected by stress proteins. They effectively exist as dormant particles until conditions improve. This ability to withstand complete dehydration had profound implications for understanding how life might persist in seemingly inhospitable environments, not just on Earth but potentially on other worlds. Environments previously considered too dry to support life, such as the Atacama Desert or regions of Mars, might harbor organisms with similar adaptations. The radiation resistance of audaxviator similarly defied conventional biological limitations. Laboratory tests exposed samples to radiation doses thousands of times higher than what would be lethal to human cells. Not only did the bacteria survive, but they showed evidence of actually utilizing the radiation as an energy source, converting damaging radiation into usable chemical energy. They possess a multi-layered radiation defense system, Elena documented in her research notes. First, physical protection through specialized cell wall structures that absorb or deflect radiation. Second, chemical protection via free radical scavenging compounds. Third, extraordinarily efficient DNA repair mechanisms that can reconstruct damaged genetic material with minimal errors. This radiation resistance had evolved as an adaptation to their natural habitat, where uranium decay in surrounding rock exposed them to constant low-level radiation. Over millions of years of isolation, this pressure had driven the development of repair mechanisms far more sophisticated than those found in surface organisms. The defensive capabilities of audaxviator extended to chemical tolerance as well. The bacteria showed remarkable resistance to heavy metals, acids, and other toxic compounds that would destroy conventional cellular structures. They accomplished this through a combination of specialized membrane transport systems that prevented toxins from entering the cell and internal detoxification mechanisms that neutralized any harmful compounds that did penetrate their defenses. They are essentially impervious to chemical attack, Richard noted during a laboratory demonstration where the bacteria continued to function in a solution containing mercury concentrations that would be immediately fatal to most life forms. They can either exclude toxins entirely or transform them into harmless compounds. This chemical resilience had practical implications beyond pure science. Bioremediation, using biological organisms to clean up environmental contaminants, represented a growing field in environmental restoration. Organisms with audaxviator's abilities to neutralize toxins could potentially transform heavily polluted sites that currently defied conventional cleanup methods. Perhaps most intriguing of all the bacteria's survival adaptations was their response to vacuum exposure. When samples were subjected to the vacuum conditions of space in simulation chambers, a significant percentage remained viable. They accomplished this feat through rapid formation of spore-like structures with multiple protective layers that prevented the loss of all cellular water and protected essential molecular machinery. They essentially create a microscopic spacesuit, Elena explained to a fascinated audience at the European Astrobiology Conference. A multilayered protective structure that maintains minimal internal pressure and prevents fatal water loss through sublimation. This vacuum resistance, combined with their radiation tolerance and ability to remain viable when completely desiccated, meant that audaxviator possessed all the traits necessary to potentially survive interplanetary transport. A key consideration in discussions of panspermia, the hypothesis that life could spread throughout the universe via meteoroids, asteroids, and other small bodies. The cumulative picture that emerged was of an organism with survival capabilities that transcended conventional biological limitations. Audaxviator did not merely endure extreme conditions. It had evolved to thrive in them, developing specialized adaptations that turned environmental challenges into opportunities. These findings prompted a reevaluation of habitability criteria not just for Earth environments but for astrobiology as well. If life could adapt to such extreme conditions on our planet, the range of potentially habitable environments elsewhere in the universe expanded dramatically. The exceptional resilience of Audaxviator raised profound questions about the historical trajectory of life on Earth and its potential distribution beyond our planet. These questions became the focus of an international research initiative led by Elena and Richard in the years following their Lake Baikal discovery. One of the most compelling hypotheses to emerge concerned the evolutionary age of these extraordinary bacteria. Conventional molecular clock analyses, which estimate the time of divergence between species based on mutation rates in certain genes, suggested something remarkable about audaxviator. The molecular evidence indicates these organisms diverged from other bacterial lineages approximately 3.8 billion years ago, Richard explained during a symposium at Cambridge University. This places their origin very near the beginning of life on Earth, possibly among the first successful cellular organisms. This extreme age would explain many of the bacteria's unusual features. They might represent a distinct evolutionary branch that diverged before many of the characteristics now considered standard for bacteria became fixed. Their unique cellular compartmentalization, for instance, could reflect an early experiment in cellular organization that predated the conventional prokaryote-eukaryote split. The ancient origin hypothesis gained further support from comparative genomic analysis. Audaxviator possessed genes with no known homologs in other living organisms but which showed striking similarities to genes reconstructed from fossil evidence of Earth's earliest microbes. They appear to have retained genetic elements that other lineages lost or significantly modified billions of years ago, Elena noted in a paper published in Nature. It is as though they preserve a genomic snapshot from life's early history on Earth. This preservation of ancient genetic elements was likely a consequence of the bacteria's isolated habitat. Without competition from other organisms or exposure to the dramatic environmental changes that drove rapid evolution on Earth's surface, audaxviator populations evolved much more slowly, retaining characteristics that disappeared elsewhere. The bacteria's extreme age and unusual evolutionary history led to what became known as the deep biosphere hypothesis. The proposal that a significant portion of Earth's biomass and biodiversity existed not on the surface but in the planet's crust, extending kilometers downward into regions previously thought devoid of life. Initial surveys supported this hypothesis. Sampling expeditions to deep mines, oil wells, and drill sites around the world frequently recovered evidence of microbial communities with similarities to audaxviator. These communities varied in composition based on local geology but shared key adaptations for surviving in high-pressure, low-oxygen, low-nutrient environments. We are discovering that the subsurface biosphere is not a rarity but a major component of Earth's living systems, Richard testified before a congressional science committee seeking funding for expanded deep biosphere research. Current estimates suggest subsurface microbial biomass may equal or exceed that of surface life. This revelation transformed ecological understanding of our planet. Earth was not merely a rocky sphere with a thin coating of life on its surface and in its oceans. Rather, it was a living planet throughout much of its outer crust, with diverse ecosystems extending kilometers beneath our feet. The implications extended to planetary habitability assessments throughout the solar system. If life could thrive deep within Earth without dependence on surface conditions, similar subsurface habitats might exist on other worlds, most notably Mars and several icy moons of Jupiter and Saturn. The surface of Mars is hostile to life as we conventionally understand it, Elena explained during a NASA consultation. But subsurface environments, protected from radiation and potentially containing liquid water, might harbor Martian versions of audaxviator, organisms completely adapted to life beneath the surface. This perspective shifted astrobiology search strategies. Rather than focusing exclusively on surface features when looking for signs of extraterrestrial life, missions increasingly incorporated technologies for subsurface exploration and sampling. The astrobiological significance of audaxviator extended beyond its implications for where life might be found to questions about how life might travel between worlds. The bacteria's extraordinary resilience to vacuum, radiation, and desiccation made it one of the few known organisms potentially capable of surviving interplanetary transport. This revitalized scientific interest in panspermia, the hypothesis that life could be distributed throughout the universe by traveling on meteors, asteroids, or comets. While most organisms would perish during the harsh conditions of space travel, audaxviator's suite of survival adaptations suggested it might endure such journeys. Laboratory simulations supported this possibility. When subjected to conditions mimicking those experienced by rocks ejected from a planetary surface into space and eventually landing on another world, a small percentage of audaxviator samples remained viable. They enter a state of profound dormancy during transit, Richard explained to an astrobiology conference. Metabolism ceases entirely, and specialized protective compounds prevent fatal damage to essential cellular components. Upon arrival in a suitable environment, they can reactivate and resume normal function. This capability raised the intriguing possibility that life might have originated only once in our solar system, or even beyond, and subsequently spread to multiple worlds through natural processes of planetary material exchange. Evidence for such exchange was well established. Martian meteorites had been discovered on Earth, ejected from Mars by asteroid impacts with sufficient force to overcome the planet's gravity. Similar processes could potentially transport Earth rocks to Mars or other bodies in the solar system. If microbes like audaxviator could survive inside these traveling rocks, life that originated on one world might establish itself on others. This would mean that life throughout our solar system, if found, might share a common ancestor, a prospect that would revolutionize our understanding of biology's distribution. These considerations led to enhanced planetary protection protocols for space missions. If Earth microbes could potentially survive interplanetary journeys, great care needed to be taken to prevent unintentional contamination of other worlds during exploration, which could both harm indigenous life and confuse scientific investigations. As research on audaxviator continued, the bacteria increasingly came to represent not just a biological curiosity but a fundamental reconsideration of life's essential parameters, distribution, and history, both on Earth and potentially beyond. The profound implications of audaxviator extended into another surprising domain, artificial intelligence and computing systems. The bacteria's information processing capabilities and resilience attracted attention from computer scientists and engineers seeking inspiration for next-generation technologies. 3 years after the Lake Baikal discovery, Elena found herself in an unexpected collaboration with the Advanced Computing Systems Laboratory at the Massachusetts Institute of Technology. The partnership began after computer scientists became intrigued by the bacteria's method of storing and processing information. Conventional computing systems separate memory and processing, explained Dr. Maya Patel, the laboratory's director, during their initial meeting. Data is stored in memory units and moved to processors for computation, what we call the von Neumann architecture. This creates a bottleneck that limits efficiency. audaxviator, however, employed a fundamentally different approach. Rather than separating storage and processing, the bacteria integrated these functions through a complex network of RNA molecules that could both store information and perform computational operations on that information simultaneously. They essentially employ a biological version of what we call neuromorphic computing, Elena noted. Information storage and processing occur in the same physical substrate, similar to how brains work rather than conventional computers. This biological computing system demonstrated remarkable efficiency. audaxviator could process environmental information, evaluate multiple response options, and implement appropriate adaptive changes while consuming minimal energy, operating on a power budget measured in picojoules rather than the watts required by electronic systems. The bacteria accomplished this feat through a network of catalytic RNA molecules, ribozymes, that could both store information in their sequence and perform computational operations through their three-dimensional folding and chemical activities. These RNA networks could reconfigure themselves in response to environmental signals, essentially reprogramming their computational architecture on demand. It is a completely different paradigm for information processing, Richard explained to the engineering team. Not binary digital computation, but analog chemical computation with massive parallelism. Inspired by these biological mechanisms, computer scientists began developing artificial systems that mimicked the bacteria's integrated storage processing architecture. These bacterial-inspired processors represented a radical departure from conventional computing approaches. Traditional computing separates hardware and software, Dr. Patel explained in a subsequent research paper. In these new systems, like in audaxviator, the hardware itself is malleable, reconfiguring in response to computational needs. Beyond its computing architecture, the bacteria's error correction mechanisms provided another source of technological inspiration. Living in radiation-intensive environments had forced audaxviator to develop sophisticated methods for detecting and repairing damage to its information storage systems, primarily its DNA. These error correction protocols operated on principles that transcended existing computational methods. Rather than simply detecting errors through checksums or redundancy, the bacteria employed predictive systems that could reconstruct lost information based on context and pattern recognition. They do not just identify that an error has occurred, Elena explained. They can often determine what the correct information should be based on contextual cues and overall information coherence. Engineers began implementing similar contextual error correction in critical systems where data integrity was paramount, such as spacecraft control systems and medical devices. These approaches proved more robust than conventional methods, particularly in high-radiation environments where traditional electronics were vulnerable to bit flips and data corruption. Perhaps the most significant technological application emerged from studying audaxviator's energy harvesting systems. The bacteria's ability to derive useful energy from minimal and diverse sources, including waste products and background radiation, inspired new approaches to power generation for small-scale electronics. Conventional power sources like batteries have fundamental limitations in terms of energy density and lifespan, noted Dr. James Chun, a materials scientist who joined the research team. These bacteria demonstrate methods for harvesting ambient energy that we had not considered viable. One particularly promising application involved medical implants, devices traditionally limited by battery life and the risks associated with replacement surgeries. Researchers developed biocompatible energy harvesting systems, inspired by audaxviator's metabolism, that could power implants indefinitely by utilizing the body's own chemistry. The first generation of these self-powered pacemakers is already in clinical trials, Richard reported at a biomedical engineering conference 4 years after the Lake Baikal expedition. They derive power from the same chemical gradients and processes that the bacteria exploit, eliminating the need for battery replacement surgeries. Beyond specific applications, audaxviator prompted a philosophical reconsideration of resilient system design. Modern technologies typically achieved reliability through redundancy, multiple backup systems that could take over when primary systems failed. The bacteria, however, demonstrated a different approach, fundamental resilience at the most basic structural level. Rather than building vulnerability into the system and then compensating with backups, these organisms build resilience into their core architecture, Elena observed. Every component is designed to function across a wide range of conditions. This principle began influencing critical infrastructure design, particularly for systems that needed to operate in extreme environments or emergency situations. From disaster response robots to deep space probes, engineers increasingly prioritized inherent resilience over complex backup systems, creating technologies that could continue functioning even when significantly damaged or placed under extreme stress. The technological implications of audaxviator illustrated a broader principle, that biological systems, shaped by billions of years of evolution, often contained solutions to engineering challenges that human designers had yet to discover. The bacteria represented not just a biological anomaly but a reservoir of design principles that could transform human technology. This perspective catalyzed the emerging field of deep biomimicry, the systematic study of extremophile organisms specifically to identify novel engineering approaches. Unlike conventional biomimicry, which often focused on readily observable natural systems, deep biomimicry sought inspiration from life's most extreme adaptations, often at the molecular or cellular level. Nature has been conducting research and development for 3.8 billion years, Richard noted in a TED Talk that garnered millions of views. The most extreme environments have produced the most innovative solutions we are only beginning to understand and apply. As technological applications of audaxviator's unique capabilities proliferated, they reinforced a central insight of the Lake Baikal discovery: that life's adaptability and resilience far exceeded conventional scientific understanding. The impossible animal that should not be alive was not merely surviving but thriving, and in the process, teaching humanity lessons that extended far beyond biology. 8 years after the Lake Baikal expedition, Elena stood at the podium of the Stockholm Concert Hall, the Nobel Prize medal for chemistry gleaming against her dark suit. The journey from that first glimpse of audaxviator to this global recognition had transformed not just her career but entire fields of scientific inquiry. The history of science is, in many ways, a history of boundaries. She began her acceptance speech. We draw lines between what is possible and impossible, between the living and non-living, between conditions that can support life and those that cannot. Desulforudis audaxviator has erased these boundaries, teaching us that life is not constrained by our limited understanding of its possibilities. In the audience, Richard nodded in agreement. Their partnership had weathered the intense scrutiny, controversy, and occasional hostility that had followed their initial publication. Now, their work was being celebrated for fundamentally altering humanity's understanding of life's parameters. The impact of audaxviator extended far beyond academic recognition. The bacteria's study had spawned new fields of research, technological applications, and philosophical reconsiderations of life's essential nature. Most significantly, it had transformed humanity's search for life beyond Earth. We once searched for extraterrestrial life by looking for conditions similar to Earth's surface, Elena continued. We sought oxygen, moderate temperatures, and liquid water. Audaxviator taught us that life can thrive without oxygen, at temperatures ranging from near freezing to well above boiling, and with minimal free water. This insight has revolutionized our approach to astrobiology. Indeed, space agencies worldwide had revised their habitability criteria and search methodologies based on the bacteria's extraordinary adaptations. Subsurface exploration had become a priority for missions to Mars, Europa, Enceladus, and other potentially habitable worlds. The recognition that life could exist in conditions previously considered hostile had exponentially expanded the range of environments under investigation. The first evidence validating this approach had come just months before the Nobel ceremony, when the Europa Subsurface Explorer mission detected chemical signatures consistent with biological activity in samples obtained from beneath the Moon's icy surface. While confirmation was still pending, the preliminary data suggested microbial communities with striking similarities to Earth's deep biosphere. If confirmed, these findings will represent the first discovery of extraterrestrial life, Elena acknowledged. Yet importantly, this life may share fundamental characteristics with organisms like audaxviator. This would suggest either that life has evolved similar solutions to similar challenges independently, or that life throughout our solar system shares a common origin. Either possibility would profoundly impact humanity's understanding of its place in the universe. If life had evolved independently on multiple worlds but converged on similar solutions, it would suggest that the parameters of biology were constrained by universal chemical and physical principles. If life shared a common origin, It would mean humanity was part of a broader biological continuity extending beyond Earth. Beyond astrobiology, audaxviator had catalyzed a fundamental reconsideration of Earth's own biological history. The traditional view of life's evolution as primarily a surface phenomenon driven by photosynthesis had given way to a more complex understanding of a multi-layered biosphere extending kilometers into the planet's crust, with distinct but interconnected evolutionary trajectories. We now recognize that what we once considered the biosphere, the thin layer of life at Earth's surface, represents only a fraction of our planet's living systems, Elena explained. The deep biosphere contains as much biomass and potentially greater biodiversity, operating on different energy sources and evolutionary principles. This recognition had practical implications for issues ranging from climate science to natural resource management. Carbon cycle models had been revised to account for the metabolic activities of subsurface microbial communities. Extraction industries now routinely assessed the biological impact of activities that disturbed deep crustal environments. Conservation biology had expanded to consider not just surface ecosystems but the largely unexplored biological communities beneath them. Perhaps most profoundly, audaxviator had transformed humanity's conceptual understanding of life itself. Traditional definitions based on specific biochemical processes or environmental requirements had given way to more flexible characterizations focused on fundamental capabilities: information processing, energy utilization, and adaptive response. Life, we now understand, is not defined by particular molecular structures or environmental conditions, Elena said, her voice resonating through the concert hall. Rather, it is defined by function, the ability to maintain complex organization, process information, harvest energy, and adapt to change. The particular mechanisms through which these functions are achieved may vary dramatically across different organisms and environments. This functional definition of life had philosophical implications that extended beyond biology. It suggested connections between biological systems and other complex, self-organizing phenomena in the universe, from atmospheric patterns to stellar formation processes. Life, in this view, represented not a categorical exception to physical law but a particular expression of universal principles of complexity and information processing. As her address concluded, Elena reflected on the journey that had brought her to this moment, from that first glimpse of translucent, bioluminescent organisms in Lake Baikal's depths to a comprehensive reevaluation of life's fundamental nature and distribution. The greatest scientific discoveries are not those that confirm what we already believe, she said, but those that compel us to reconsider our most basic assumptions. Audaxviator has taught us that life is not constrained by the boundaries we imagine for it. It adapts, innovates, and persists in conditions we once thought impossible. She paused, looking out at the assembled scientists, dignitaries, and students. The impossible animal that should not be alive has shown us that impossibility often reflects the limitations not of nature, but of our understanding. As we continue exploring our world and others, we must remain open to life's extraordinary capacity for surprise. As Elena stepped away from the podium to thunderous applause, screens behind her displayed microscope images of audaxviator, the unlikely organism that had reshaped humanity's understanding of life's possibilities, on Earth and beyond. In the decade following the Nobel recognition, research on audaxviator and related organisms accelerated, revealing ever more extraordinary capabilities and applications. The bacteria study spawned interdisciplinary collaborations between fields that had traditionally operated in isolation: astrobiology and computer science, evolutionary biology and materials engineering, ecology and quantum physics. These collaborations produced insights that transcended traditional disciplinary boundaries. Quantum biologists discovered that audaxviator utilized quantum coherence effects for energy transfer in its specialized organelles, maintaining quantum states at temperatures far higher than thought possible. These findings revolutionized quantum computing approaches, leading to systems that could operate at room temperature rather than requiring extreme cooling. Materials scientists, inspired by the bacteria's structural adaptations to extreme pressure, developed new classes of metamaterials with unprecedented strength-to-weight ratios. These materials found applications in everything from deep-sea research vessels to spacecraft components, enabling exploration of environments previously inaccessible due to material limitations. Medical researchers identified compounds produced by audaxviator with extraordinary protective properties against cellular damage. These compounds, when synthesized and adapted for human physiology, showed promise in treating conditions ranging from radiation exposure to neurodegenerative diseases characterized by cellular stress. Perhaps most significantly, the bacteria catalyzed a philosophical reconsideration of humanity's relationship with the rest of Earth's biosphere. The recognition that complex, highly adapted life forms had existed for billions of years in complete isolation from human activity prompted a more humble assessment of humanity's place in Earth's biological systems. We are not the culmination of Earth's evolutionary journey, Richard reflected in a widely circulated essay on the 10th anniversary of the Lake Baikal discovery. We are one expression of life's possibilities, existing alongside countless others, many of which remain unknown to us. This perspective fostered a more integrated approach to environmental management, conservation biology, and sustainable development. Rather than seeing humanity as separate from or dominant over natural systems, policy increasingly recognized the interconnectedness of all Earth's biological communities, from the surface to the deep subsurface. As Elena and Richard approached the end of their scientific careers, they could look back on a discovery that had not merely added to human knowledge but fundamentally transformed it. The impossible animal that should not be alive had rewritten textbooks, spawned new technologies, and altered humanity's understanding of life's essential nature. In her final public lecture at the American Museum of Natural History, Elena addressed a packed auditorium of scientists, students, and citizens fascinated by the story of audaxviator. Her closing remarks captured the profound implications of their discovery. Before we found these extraordinary organisms, we thought we understood the boundaries of life, what it required, how it operated, where it could exist. We drew neat categories and defined clear limitations. Audaxviator taught us that life transcends our categories and limitations. It finds a way to persist and thrive in conditions we once thought impossible. She displayed a final image on the screen behind her. Not a microscope photograph or data visualization, but a simple artistic rendering of Earth. The planet appeared not as the familiar blue marble of surface imagery, but as a translucent sphere revealing multiple layers of biological activity, from the atmosphere to the deep crust. "This is our true biosphere," she said. "Not just the thin shell of photosynthetic life we've long studied, but a living planet, permeated with life operating on different principles, energy sources, and timescales. Understanding this complex, multilayered living system is the great scientific challenge of coming generations." As Elena concluded her lecture, the audience rose in standing ovation, not merely for her and Richard's achievements, but for the extraordinary organism that had transformed humanity's understanding of life itself. Desulforudis audaxviator, the impossible animal that should not be alive, had opened doors to new scientific frontiers that would be explored for generations to come. The impossible had become not merely possible, but a source of endless discovery and wonder.